we are going to discuss about the diseases in the field crops and also horticultural crops. So, in this topic, generally in the diseases, there are three types of the diseases are there. One it is the fungal disease, the second one is the bacterial diseases, and also the third one is the viral diseases. In addition to these three diseases, we will also explain a little bit about the nematodes which are causing the huge yield losses. But today we will discuss about only the rice diseases. So after that one, that means after next class onwards, we will discuss about one topic. We will take the one topic that is either it is the cereal or whether it is the pulse crops. So total diseases we will discuss according to the whether it is the cereal crops or whether it is a pulse crop. Based on that one, we will discuss next class onwards. But today we will discuss about only
Hello, sir. Sir, please continue.
Okay. Yes, I will continue that one.
Okay.
Okay. Good evening, students. Today's our topic is rice diseases. In rice diseases, there are a lot of diseases are present in our country. Among the diseases, blast, brown spot, narrow brown spot, sheath blight, sheath rot disease, stem rot, false smut, bacterial leaf blight, bacterial leaf streak, string growth, rice grassistant virus, and all of these are the most important diseases throughout our country. But here, now we will discuss about some of the important diseases in the rice crop. Now, let us start the one of the most important disease, that is nothing but blast disease. So when it is first time identified or noticed, so the disease blast it was recorded in China in the year 1637. So later spread to the
Hello, sir. You are not audible, sir.
The small interruption. So the disease, it was first time noticed in the China. Later, it was spread to the neighboring country and also Japan. In addition to these two diseases, the disease, it was also noticed in European country, that is in Italy. In that Italy, the disease, it was simply called as a brucine. So it was noticed in the year 1828. But in our country, first time it was recorded in Tanjavur district of the Tamil Nadu in the year 1980. So what is the need of to study about the importance of all these things, the background of that disease? That means generally for any disease, first of all, we have to know about the origin of that particular disease, generally where it is originated or where it is initiated, later how it is spread to the throughout the world. So here we have to remember one thing in that background or history of the particular blast disease is nothing but first time it was noticed. Where the blast disease it was noticed in the which country? Either China, Japan, or something else. Definitely, in examination point of view, they will ask the type of question. So here you should simply blindly remember that one. First time it was recorded in China country. In Italy, what is the name of that particular disease of the blast? Brucini or Cresset or something else. So you should remember like that manner. Okay, we don't want to go to the deep into that particular subject. In our examination point of view, what type of the things are required or what type of the points you have to remember, that is the important for you. So here in blast disease, you should remember only when the disease it was first time noticed. China. What is the name of the blast disease in Italy? That is Brucine. In our country, when the disease it was noticed in the year 1918, in which part? That is nothing but Tanjavur district in Tamil Nadu. These points are very, very important according to your examination point of view. So now let us come to the economic importance. Economic importance, the yield losses. That means what is the importance of that particular disease, whether it is beneficial or harmful. Definitely, the disease means mostly at 99%, it is harmful. So if it is harmful means what is the need to study about that one? Because of it reduces the yield. So how much yield losses it causes? Nearly it causes the 30 to the 61%. It depends upon the stage of the infection. That means if it comes early in the stage means it causes more yield losses. If the disease it appears later in the stage of the crop means it causes the somewhat little bit less than 60%. Sometimes in severe cases it causes the yield losses up to 80%. That means whenever the disease cycle or whenever the secondary spread of the disease it is rapidly moving, rapidly spreading, at the time it causes the more yield losses. That means it completely depends upon the secondary spread of the disease. If the secondary spread of the disease it rapidly is spreading, means definitely it causes the more yield losses. If the spread of the disease, if it takes the very less time means definitely it causes the more yield losses. Sometimes even it causes the 80% of the yield losses also. Whenever the favorable conditions are present, whenever there is a virulent strain is present in the field, that all of these factors are very, very important for the disease severity or for development of the disease. Now let us come to the disease symptoms. What type of symptoms are produced by the pathogen? So here pathogen, it is the Pyracularia varicella. You should remember that one. So now let us come to the disease diagram. So the pathogen for this one is the Pyracularia varicella. The perfect stage of blast disease is the Magnopatha gracia. You should remember this one. Which one is the anamorph stage? Which one is the teleomorph stage? So teleomorph stage or sexual stage or perfect stage. All of these are the similar words. So you should remember that one. Magnopatha gracia is a teleomorph or sexual stage or perfect stage of the blast disease. That means perfect state of the particular pathogen. So generally, we will call it as a Pyracloria varizae is the causal agent for the blast disease. So what type of symptoms are produced by that one? So here the pathogen, it causes the two different types of the symptoms from the plants. One, it is the diamond-shaped lesion, or second one is the spindle-shaped lesions. So either of the one are produced, or sometimes both types of the lesions are produced by the Pyracloria varizae on the crop. So if you look at the left side diagram, it simply represents the formation of the diamond-shaped lesion. The right side diagram simply represents the spindle-shaped lesion. So the center of the lesion, it is the grayish white in color, which is surrounded or bordered by the reddish brown color border. If you look at the central diagram, it has given the clear-cut diamond-shaped lesion. But here we have there is not possible to see the grayish white colored border. Here the total lesion, it is in the reddish brown in color. You should remember that one. Mostly, whatever we are talking about the shapes, all the leaf spots, they will not be given the same type of the lesion. But out of the 10 or 20 leaf spots lesion, definitely some of the leaf spots, either 2 or 3 or 4 lesions, they will give this type of lesion, either diamond shape or spindle shape. Based on that one, we can identify the disease in the field. It is caused by the Pyracloria varizae pathogen. The disease, it is the blast disease. Okay, you should remember that one. Because of, in your examination point of view, there is a chances for asking one question related to the symptoms shape based on the symptom shapes. The diamond-shaped lesions are formed due to the dash. And there's the Pyracloria, Helminthosporium, and so on so on. Zactinius solani, like this manner. Because of, you should remember this one. Next one. So in addition to earlier in this slide, all of these lesions are formed on the leaf blades. There are different types of the blast diseases are there based on the which part it is produced. For example, if the lesions are formed on the leaf blade, means that is nothing but leaf blast disease. Okay, you should remember that one. In addition to the leaf blast disease, there is also nodal blast disease there. Nodal blast disease means the lesions are formed at the nodal region, which are blackish in color, and also they are irregular in shape. Here, at the nodal portion, there is a formation of the irregular shaped black colored lesions are formed, which are also elongated in shape. That means if you look at that one closely, it gives some black color discoloration. We can't identify that one exact shape because of they are very small in size when you compare with the leaf blade. Not only for this one, for any disease, if the lesions are formed on the stem portion or branches, it can't give any circular or oval shaped lesions. Simply they are elongated lesions. For any disease, you should remember that one. Now let us come to the neck blast. So neck blast, panicle blast, neck rot, all of these are the synonyms for the neck blast disease. So what is the neck blast reason? So here, the panicle neck, it is completely infected with the blast pathogen, that is Pyracloria varizae. Without that neck portion, it is infected with the Pyracloria, definitely there is no formation of the grain filling. Whenever the grain filling, it is not possible. Simply, there is a chances for the development of trophic grains. You should remember that one. If the disease, it was started to initiate the disease on the neck portion after panicle emergence means there is only few grains will be infected. That means few grains will be converted into the trophic grains. That means whenever the disease it occurs in early stages, early panicle initiation stages means most of the grains they will become as a trophic grains. If the disease or if the pathogen it attacks after emergence of the panicle means only some of the grains will be changed into the trophic grains. So trophic grains means there is no formation of the or fulfillment of the grain. So how could you confirm that particular disease as a blast disease? So the lesions are elongated and also they pointed at each end. That is the confirmation for the identification of the disease. So here we are talking about the lesions, means the lesions which are formed on the leaf blade. The lesions which are formed on the nodal region or neck region, it is not completely gives the clear-cut information regarding that particular blast disease. If the disease it occurs on the leaf blades, at that condition on the leaf blades, we can identify that one as a whether it is elongated or not. If they are elongated, if the pointed end is present, means we can say that one, we will confirm that one as a blast disease. You should remember that one. So, is there any type of similar symptoms are produced on the rice plant meat? Yes, definitely. There is also possibility to give the confusion with the some of the diseases. One, it is the suppose whenever the disease it is initial stages, most of the lesions are they are very small in size, they are circular or small in oval in shape. During that time, for this type of the symptoms are produced by the both the brown spot and also leaf blast diseases. So during the early stages of the disease symptoms of the brown spot and also blast disease, we can't differentiate such diseases. Whenever the disease it advances, at the time we can simply differentiate the brown spot and also leaf blast disease. But in earlier stages, it will give the little bit confusion among these two, between these two diseases, whether it is a blast or whether it is a brown spot. At that time, it is not easy to differentiate that one. But in later stages, we can identify that one. Next, another important similar symptoms are the white earhead formation. So due to attack of the stem borer, there is also possibility of the formation of the white earhead symptoms. That is chaffy grain formation. So here there is a two different types of the symptoms are produced on the same host plant, which are similar to the blast disease. One it is the pinhead sized brown lesion, and also second one is the white earhead formation. So you should remember these points. How could you confirm that one? Each type of the symptoms are produced along with the blast disease. So what are the collateral hosts for the blast disease? That means, so here the pathogen it causes the symptoms on the rice plant. But what is the need of the collateral host? So collateral host means whenever the main host is not available at the time, the pathogen it has to survive during that period. That is called off season. So the wild hosts or the collateral hosts they are helpful for the continuation of the life cycle of the pathogen. So the collateral hosts, they are helpful. What type of the collateral hosts are helpful for the development of the blast disease? One, it is the panicum repens, Diphtheria marginator, EC hexander, echinoclover, Ceteria intermedia. All of these are the some of the collateral hosts for the blast disease. That means even though the rice plants are absent, the pathogen it can survive still during off-season due to the presence of the collateral hosts. You should remember that one. So these are the diseases and also what type of the collateral hosts are the helpful for the survival of the pathogen. Next one is the pathogen biology. So here the pathogen it produces some of the toxins. They are alpha picolinic acid, pyrrolidine, and also pyroxolol. All of these three are the some of the toxins they are produced by the Pyrrolidine or is a pathogen. You should remember only the toxin name. The toxins they will be discussed in the toxin classification chapters. So next one is the conidia. What type of conidia is produced by the Pyrrolidine pathogen? So the Pyrrolidine pathogen it produces the pyriform to the obclavate conidia. Pyriform to the obclavate conidia, or somewhat it is a top-shaped conidia. Top-shaped conidia, which is highlighted to the pale olive green in color. So here, highlight to pale olive green color, it is that means initial stages. Most of the conidiophores and also conidia are vegetative mycelium. Mostly they are initially highlighted in color. That means there is no color for their structures. Later, as the disease advances, as the pathogen it continues the growth after a certain period, the hyaline mycelium it simply converts into the some different colors. Either it is the olive green or reddish brown or brown color, whatever it may be. So here, in your examination point of view, you should remember the shape of the conidia in blast disease. That is pyriform or obclavate or top-shaped conidia. It will be produced by the Pyrrolidine pathogen. Usually, they are three-celled structures. That means only two sets are present. Along with that one, it attaches the broader base by a hilum. You should remember that one. So a small project which come out from the base of the conidia it is called hilum. So hilum it is also important for the identification of the pathogen based on the conidial structure. So Pyrrolidine, it was first time detected by the Cera scientists in Italy in the year 1891. You should also remember that one. Who was the first time identified the Pyrrolidine pathogen? Okay, you should remember this one also. Next one is the disease cycle. That means how the pathogen it continues its life cycle. It continues its life cycle by the host plants. The disease cycle is nothing but there are two different sources are available. One it is the primary source of the infection. The second one is the secondary source of the infection. The primary source of the infection or the primary source of the inoculum, it is the mycelium and also conidia which are present in the infected straw or infected plant debris and also seeds. That means so here the seeds they may also be infected with the blast pathogen. So whenever the crop it reaches to the harvesting stage, at that time, whenever the leftover field it contains the mycelium and also conidia in that debris, they are helpful for the initiation of the disease for the next season crop. The secondary infection means whenever how the pathogen it rapidly progresses in the standing crop. For example, if any field it is infected with the pathogen, means initially some of the plants only infected. That means very negligible plants. That means either 10 plants or 5 plants, they may be infected with the pathogen during the earlier stages. But within the short period, within the 7 days or 10 days or 15 days, the disease it will be rapidly spreading. That means in the standing crop, how the primary infection it gives the rapid spread of the disease. That means by the wind-borne conidia. That means whatever the conidia it is produced on the primary infected plants, they will be rapidly spread to the different directions based on the wind, based on the rain, all of the weather factors, they are helpful for the spreading of the disease. So you should remember that one, wind-borne conidia. The conidia, it will be helpful for the spreading of the disease. So all of these are the present, but there is it lacks the it needs the some other factors. That means it requires the favorable factors. Whenever the favorable factors are absent, even though the host and also pathogen is present, means definitely that disease it will not be developed at a particular point of the time and also in a particular field. That means the field it is ready, the pathogen it is ready, it is available. That means the pathogen it is also present in that locality. But the favorable factors are not available means definitely the disease it will not be initiated. That means here it requires the third factor. It is the weather factors. They are also required for the development of the disease. So what type of the factors are required for the initiation of the disease? Number one is the application of the excessive dosages of the nitrogenous fertilizers. That means here the farmers they will apply the fertilizers beyond the optimum level, beyond the optimum dosages. What happens if they apply the excess level? Means generally the nitrogenous fertilizers, if you apply the any nitrogenous fertilizers just like urea, definitely the vegetative growth stage it will also be increased. Whenever the vegetative stage is increased, at the time there is a possibility for the development of the disease by the pathogen. That means the time it will be increased for the attacking of the pathogen. So that is the thing, and also intermittent drizzles. That means if the rain it is there, the rain showers are present, they are also helpful for the development of the disease. Cloudy weather, high relative humidity. That means more than 90% relative humidity. It is favorable for the development of the blast disease. Low night temperature. That means 15 to 20 degrees centigrade is highly favorable night temperature for the development of the conidial production. More number of the rainy days. That means at least it requires the 2 days of the rainy days. If more than 2 days of the rain means it is also helpful for the rapid spread of the disease. Longer duration of the dew. So dew means the leaf wetness. Whenever the dew it is present on the leaf blade, that will also be helpful. On which way it is helpful? Whenever the dew it is present on the leaf blades, generally the conidias are produced during the night time of the during night time. Whenever the conidia it is produced during the night time, definitely they will spread to the remaining areas. Whenever they are spread to the remaining areas, they are disseminated to the some other places. Definitely, by force, the conidia they will be landed on the leaf blade and also different aerial parts. But whenever the wet it is available, whenever the dew it is present on the leaf blades, they are helpful for the germination of the spore. That is the thing. Next one is the slow wind movement, also availability of the collateral host. Already we have told about the role of the collateral host. So all these factors are helpful for the development of the blast disease. Why we have mentioned all these things means most of the more or less this type of the favorable factors are required for the many diseases. That's why now we have discussed about the favorable factors. That means most of the diseases they require the low temperatures and also high relative humidity, cloudy weather, intermittent drizzles, and also excessive application of the nitrogenous fertilizers. So all of these are the mostly favorable factors for the many diseases in the field crops and also horticultural crops. Okay. Next one is the forecasting model. That means simply the name it denotes forecasting. Means before appearance of the disease, how could you identify that one? Some of the scientists they have developed the forecasting model. That means before appearance of the disease, based on the weather factors, the scientists they have developed the many models. Among this one, BLAST it is the one of the forecasting model which was developed in Japan. So this is the first leaf blast model. You should remember the name it is the blast disease, but the name of the forecasting model it is also a blast disease. In addition to the blast forecasting model, some of the models are also developed by the different countries. So among this one, Pyriclaria is also one of the forecasting model. PyriView it is also one of the forecasting model. Blast Aim and also P-Blast. All of these are the forecasting models developed for the blast disease management. So in addition to all these models, in our country also developed one forecasting model that is nothing but EPIPLA. EPIPLA it is also one of the forecasting model which is developed in our country. EPIPLA means nothing but epidemiology of the blast disease. They have taken in epidemiology, blast disease, they have taken the BLA. So that's why they have given the EPIBLA for the forecasting model. So they have done the experiment in the rice cultivars IR50, IR20 varieties in Madras University. So Manibhushan Rao and also along with the Manibhushan Rao, some of the scientists they are also helpful for the development of the EPIBLA rice blast model in the year 1991 in Madras University. So Manibhushan Rao he developed the forecasting model in our country from University of the Madras. The name of the blast model is the EPIBLA, and also they have conducted the experiments on IR50 and also IR20 varieties. So among this one, we should remember the forecast model developed in our country. They will give the choices Pyriclaria, PyriView, EPIBLA, and also P-Blast. So you have to select the EPIBLA. It is developed from our country. So management. So in management, there are different methods of this. The first one is we have to select the resistant variety. So the resistant varieties, they are different for the different states. So you should remember only some of the resistant varieties according to your state, and also some of the resistant varieties they are common for the many states. Next one is the seed treatment. So seed treatment for the blast disease is the Trichoderma viridae. Whenever Trichoderma viridae is applied as a seed treatment means you should remember that is only 4 grams per the kg of the seed. If you apply Pseudomonas fluorescens as the seed treatment means you should apply 10 grams per kg of the seed. You should remember these points whenever the management practices are there. So next one is the spraying of the chemical. Whenever the field it is completely infected with a particular disease means definitely we have to move for the spraying of the fungicide. So here, what type of the fungicide is required for the management of the blast disease? That is Edifenphos at the rate of the 0.1%. We should remember this one. So here we have given some of the examples for the management of the blast disease. So pyraclostrobin, that is the one of the fungicide. It is only used for the blast disease. Tricyclazole is also used for the blast disease. Cosgamycin, isoprotheolone, all of these are the fungicides. They can be used only for the management of the blast disease. So here we should remember, along with the Edifenphos, we should also remember these fungicides, because of these fungicides are also used for the management of the blast disease. For example, if the field it is infected with the both leaf blast and also neck blast, means at the time we should go for the combination fungicide, that is nothing but picoxystrobin and also tricyclazole. Picoxystrobin and tricyclazole. Okay. In addition to that one, there is, we have written in the bracket only for the blast and also sheath blight disease. That means here hexaconazole and validamycin combination fungicide is required for the management of these two diseases. So there is a possibility to ask a question. The field it is infected with the blast disease and also sheath blight disease is present. What is the recommended fungicide? What is the recommended fungicide for the management of both the diseases? At the time, there is a chances to ask to give all the multiple choices. Edifenphos, pyraclostrobin, also they will give the one or two combination fungicides. In that time, you have to pick up the combination fungicide, hexaconazole and also validamycin. So next, another important disease in the rice crop, it is the brown spot. It is caused by the Helminthosporium variae or Drusilla variae. The sexual stage for the brown spot disease is the Cochleobolus myopenus. Cochleobolus myopenus. Okay. Next one is what is the, so here the brown spot, it is also one of the main principal cause of the Bengal famine in the year 1942 to the 43 years. So in that year, the brown spot, it caused the Bengal famine. So famine means it causes the huge yield losses, and also many people they starved due to lack of the food materials, especially rice crop. And also nearly more than 50,000 people they have died due to the non-availability of the rice crop. Okay, now anyhow, what is the main reason for the development of the Bengal famine? The disease it appeared in West Bengal and also now Bangladesh region. That means in that area, the Bengal famine happened. But what is the main reason for that one? Means the favorable conditions for the development of the disease was continuously maintained up to 30 days of the period whenever the crop it is in standing position. Generally, for the development of the disease and also for the rapid spread of the disease, generally 5 to 10 days are present for the initiation of the disease and also for the development of the disease. But whenever the Bengal famine occurred during that period, the highly favorable conditions are continuously maintained up to 1 month of the time. Whenever the 30 days of the time it is present, especially for the favorable conditions for the development of the brown spot pathogen Helminthosporium, during that time, the pathogen it rapidly spreaded by multiplication of the pathogen in millions of the times. That means, suppose if the one spot it produced the 100 spores once, the 100 spores they will fall in on the some other places whenever the crop it is available. all the spores they are started to germinate, and also they produce again leaf spots disease. We know that completely the total leaf green area, it is occupied with the brown spots. What happens? Totally the photosynthetic area, it will be reduced. We know the total photosynthetic area, it is reduced. There is no chances for the development of the grains. There is no chances for the development of the panicle initiation. So that type of situation happened in the year 1942 to the 1943 year. So who was the first scientist identified the brown spot disease? Means Sundar Raman from the Madras in the year 1919. He was the first person identified the brown spot disease from the Madras area from Tamil Nadu state. So here also we should remember this one also. So let us see the diagram. So initially they are very small in size, and also the spots they appear as a sesamum seeds. That's why it is called sesamum leaf spot. And also the disease, it is caused by the helminthosporium. So that's why the name was also given as a helminthosporious. So helminthosporious, sesame leaf spot, brown spot, all of these are the synonym for the brown spot disease. Now you should remember when the disease it attacks. So the disease or the pathogen, it attacks from seedling stage to the milky stage of the crop. That means in field conditions. So here there is a chances for the development of the disease from the seedling stage. That means from the nursery stage to the up to field conditions. So what type of spots are affected with the pathogen? So in initial stages, it also attacks the coreopter, leaf blades of the crop, leaf sheath, and also blooms. That means the reproductive parts, they are also infected. That means most of the aerial parts are infected with the pathogen. So what type of the lesions are formed? They are initially, they are very small, minute brown colored dots are formed on the leaf blades. Later, they become circular, oval, or cylindrical in shape. Mostly, they are either circular or oval shaped lesions, and also they are dark brown to the purple brown in color. If you look at that diagram, they are dark brown or purple color lesions are formed on the leaf blade. Leaf blade, mostly they are light brown or grayish center, which is surrounded by the reddish brown margin. It is mainly due to the production of the toxins. Dark brown or black spots, they are also appears on the blooms, which contains the large number of the conidiophores and also conidia of the fungi. In addition to the leaf blade, the pathogen, it may also attack the grains of the rice crop. So on the rice grains, you can see the formation of the black spots. Black spots. So the disease, it may also associated with one of the physiological disorder in the Japan, that is nothing but Akiyoshi disease. So Akiyoshi is one of the physiological disorder. You should remember that one. The brown spot disease is caused by the helminthosporium, but the Akiyoshi, it is one of the physiological disorder. But most of the times, the brown spot disease, it will associate with the one of the physiological disorder, especially in Japan country. What is the main reason for the development of the Akiyoshi disease in rice means hydrogen sulfide toxicity. Whenever the hydrogen sulfide it is present in excess level, it causes the black crown and also root rot in the rice crop. If you pull out that plant, the total roots, they are simply converted into the black to the brown color, black to brown in color. So here we should remember APOC, it is the physiological disorder. Next, another important thing is the pathogen. It will also associate the disease. It will also associate with one of the physiological disorder, that is potassium deficiency. So due to the potassium deficiency, the rusty brown spots that may also be formed on the older leaves of the crop. During that time, in initial stages, there is also, it gives some of the little bit confusion between the potassium deficiency and also brown spot leaf spots, brown spot lesions. So it will also be give some little bit confusion between the brown spot and also potassium deficiency. So what is the requirement of the potassium? Means potassium, it is also helpful for the disease and also drought resistance in the plant. You should remember that one. Whenever the potassium deficiency are also there is also possibility for the development of the brown spot also. So how could you confirm that brown spot disease? That is, brown spots lesions are mostly, they are circular to oval in shape, and also they are small in size whenever you compare with the blast disease. Okay, we should remember that one. Another important point is the mycelial mats, which are black and also velvety in color, which are formed on the glooms of the affected spikelets. If you take the spikelet which is infected with the disease of the brown spot, means you can easily identify that one by the formation of the velvety mycelial mat, which is black in color. If you touch that leaf, if you touch with that spot, it gives the mycelial touch. That means small powder-like structure. It will attach it to our finger. It is mainly due to the presence of the velvety mycelial mats on the globes. So similar type of the symptoms. Already we have told that one, the brown spots, they will also give some of the confusion with the blast disease. But blast disease, it will be identified by some of the important points. That is, already we have discussed in blast disease. They are elongated and also they contain the pointed ends. But here in brown spot, there is no pointed ends. They are simply circular or oval shaped lesions. You should remember that one also. Next one is the brown spot. It is the structure. It is nothing but the conidiophore and also conidia of the pathogen. The conidia usually they are bulged in the center. That means they are wider in the center and also tapering towards the end portions. They are tapering. That means they become very small in size when you compare with the middle portion cells of the conidia. Mostly they are olive green to the golden brown in color. 6 to 4 such are present in that conidia. You should remember the shape of the Helminthosporium conidia. The perfecture of the crop is the diabenus, that one. And also it produces some of the phytotoxins, ophiobolin A, ophiobolin B, and also ophiobolin I. Among these phytotoxins, ophiobolin A, it is the most toxic one. That means if you compare with the remaining phytotoxins which are produced by the Helminthosporium, ophiobolin A, it is the toxic one. So this results in breakdown of the protein fragment of the cell wall, resulting in partial disruption of the integrity of the cell. That means what is the role of the phytotoxins? Phytotoxins of the Helminthosporium means it simply break down the protein fragments, the proteins which are present in the cell wall. Whenever the proteins are present in the cell wall, if they are break down by any phytotoxin, means the cell it become very loose in position. They are not compact, they will become very loose. Whenever the cell wall it become very loose in conditions, means there is more possibility of the penetration of the pathogen, or sometimes whenever the pathogen it penetrates easily into the inside of the host cells, definitely it can occupy the most of the area, and also it will drag the nutrients. So that is the thing. That means if anything it is in compact position, if it becomes very loose, it becomes very thin or very loose, means definitely there is a more chances for the penetration of the pathogen into the inside of the host cells. So here the toxins, they are also playing the important key role for the breakdown of the proteins which are present in the cell wall of the host. So disease cycle. So if you come to the disease cycle, it is the externally seed-borne pathogen. That means that only the blooms are infected with the pathogen. Whenever the infected seeds, they are utilize it for the sowing purpose, at the time the disease it will be initiated in the next season crop. You should remember that one. Along with that, seeds, the collateral hosts, whatever we have discussed in the blast disease, they are also helpful for the survival of the pathogen during the off-season period. You should remember that one. The favorable conditions, more or less similar to the brown spot disease. And also management, you should also go for some of the tolerant varieties, and also crop rotation, adjustment of the planting time. All of these are some of the cultural practices are required for the management of the blast disease. Finally, you should go for the spray with the fungicide, that is mancozeb, at the rate of the 0.2% once after flowering, and also second spray at the milky stage of the crop period. So if you come to the chemical treatment, Edifen POS is also used for the both the blast and also sometimes for the brown spot disease. Along with this one, hexaconazole, also tebuconazole plus trifloxystrobin. These are some of the fungicides are used for the brown spot disease management. Generally, the farmers, they are used hexaconazole as a fungicide for the management of the brown spot disease. In addition to the hexaconazole, there is also presence of the combination fungicides. Carbendazim plus mancozeb, it is also one of the combination fungicide. Hexaconazole plus gineb, it is also one of the combination fungicide. Tebuconazole plus trifloxystrobin, this is also one of the combination fungicides. Suppose if you apply this combination fungicide, it will be better for the management of the disease. Otherwise, if you continuously or regularly, if you spray the only one type of the fungicide, means definitely the pathogens, they become resistant against the particular fungicide. So that's why you have to remember some of the combination fungicides also for your examination point of view, and also for your future purposes. That means if any farmer, if he wants to spray the fungicide regarding the diseases, means should also remember this fungicide also. So narrow brown spot and also sheath blight, sheath rot, all of these diseases are present. I will discuss all of these diseases in next class, but my classes initially they are very slow. Once we adjusted for the diseases for any class after a certain period, that means after 2 to 3 classes, I will continue, and also I will complete all the syllabus which are related to the plant pathology. It is not a problem. But why I have discussed very slowly for this disease means because of you have to remember the name of the disease, what is the perfect stage, what is the imperfect stage of the particular disease, and also what is the main symptom. How could you identify the particular disease is the important. Also, along with that one, what is the major important management practice for the particular disease? One, it is the resistant variety. The second one is the fungicidal spray. In addition to these two, is there any special management practices management practice is present for that particular disease. These 3 points will be covered in the management practices. So you should remember like that manner. Otherwise, if once the disease charter it is completed means if you want to remember all these things means it is difficult to you. So first of all, you should remember the disease name. That means if you select the any crop, for example, today we have discussed only 2 diseases. Next class we will discuss remaining diseases. If you take the any crop, for example, rice disease means you have to remember all the diseases names. Blast. How could you identify the blast? What is the causal organism for the blast disease? What is the perfect stage for that particular pathogen? What is the main management practice for that one? When the disease it appears, is there any similar type of the disease is present in the rice crop? How could you differentiate that particular rice blast from the some other diseases? You should remember like that manner. Otherwise, it is difficult to identify all these things. So today's topic is completed. Do you have any doubts regarding this one? Please ask me. So here some of the persons they are asking about the books. Generally, for the diseases, are you are asking for the total plant pathology which is covered in the plant sciences? Or for any for total plant pathology? That means in plant pathology, generally the fungi H.D. Dubey book it is helpful for you. That means it will give the some of the basic knowledge regarding to the fungi. That means plant pathology, especially that one comes under the mycology. The second one is the diseases. Especially, you don't need to study the all the diseases which are covered in your UG. That means, for example, if in rice crop, generally, what are the major diseases they are present in the rice crop? They will ask like that one. And also, what is the causal organism for the rice blast disease? They will ask only simple questions. If you go deep into that subject means, after completion of your preparation, it will give a little bit confusion. What is the correct answer for that one? So first of all, you should purchase the H.C. Dubey book regarding to the fungi, bacteria, and also viruses. That is the very small book. It will be helpful for you to understand the first of all what is the fungi, what is the mycology, what is the classification for that one. That means first of all you have to know about the pathogen where it comes under or which division or which subdivision or which phylum. First of all, you have to know about that one. In that phylum, what are the main features? What are the main characteristics comes under particular phylum? What are the examples are present in that phylum? You have to know about all these things. After that only, you can easily understand all these things. Otherwise, it is the difficult thing. So, plant pathology, it is not yet difficult for you. Suppose if you prepare in proper way. First of all, you have to know about the textbook H.C. Dubé, Fungi, Bacteria, and also Viruses. That is a very, very small book. It will be helpful to you. Or sometimes, if you want to exact meaning, what is that particular thing? That means, what is the mycelium? What is the primary mycelium? What is the mycelial modification? If you want to completely regarding that one means, actually, myself, I have also written the one textbook related to the mycology. That is also very, very small book, handbook mycology. That is also one of the book. It is also helpful to the students. What is the mycelium? What is the, that means handbook on an introductory mycology. NP Ishwar Reddy and also myself, both of us have written that textbook. It will also helpful to the students. That means what is the meaning of that one? If you want to know about that one very clear cutly, at that time it is easy to understand that one. Why I have written that textbook means myself also, whenever I was in PG level, at the time also I have got a lot of doubts regarding that one. Why the scientist, why the professor, he says something, sometimes mycelium, sometimes he mentioned about the hyphae, sometimes he will mention so on, so on thing. Exactly what is the meaning for that one? When they utilize that word, when they use that word, all of these are the some of the doubts regarding that particular subject, pathology. At that time, I have got a lot of doubts regarding that one. For my clarification, I have written one of the notes regarding to the fundamentals of the pathology. So that one, it was completely concluded and also added that one. And finally, we have produced that book, Handbook on an Introductory Mycology. In addition to the HCP book, if it is available, you should also purchase that one. That is published by the Kalyani Publications. It will give the some of the clarification regarding the plant pathology, especially in plant sciences. There is a possibility to ask the questions from the 3 to 4 sections. One, it is the classification of the mycology. That means the fundamentals regarding to the mycology. The second one is the diseases. The third one is the fungicides. That means management practices. What are the principles for the management of the disease? So in these 3 sectors, there is more possibility to ask the questions regarding to the plant pathology. Is there indoors? Okay, right. So here, the thing he asked that I'm reading book thoroughly. Sir, will you teach us about fundamentals of pathology or only disease? No, 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 no. Yeah, I will discuss about the fundamentals of the pathology and also diseases. But now, why I have started the diseases means at least you have to remember the name of the pathogens, and also name of the, how could you identify that one? For example, pyraclaria. Pyraclaria. It causes the disease, blast disease. How could you identify the pyraclaria? Based on the conidial structure. What is the shape of the conidia? Conidia, it is the top shape or pyriform, and also which contains the two septa. These are the things related to the pyraclaria pathogen. So whenever the diseases we are discussing about that one, at the time, definitely we have to know about the name of the causal organisms. If you know about the causal organism names and also the characteristics of the causal organism, at the time, if it is completed later, we will discuss about all these names of the causal organisms, where they will come through that one. That means which phylum all of organisms are kept. That means at the time we will study about all the characteristic features of that particular pathogen. Definitely, I will teach both the things, no doubt at all. Is there any doubt regarding that one? Don't worry about that one. Definitely, pathology means it gives some of the little bit doubts and also confusion. But after completion of that one, that means, for example, day after tomorrow or whenever the time it is there for pathology, I will discuss about some of the diseases regarding to some of the crops. After completion of that one, according to the syllabus, they told that one rust diseases, smut diseases, and also some of the diseases, leaf spot diseases. Finally, I will conclude the one notes where the smut diseases occur, which crop that disease is infected. That means, for example, rust disease. The rust disease, it is present in groundnut, in sunflower, wheat, and also sorghum, bajra. So I will give the one notes regarding to that one. That means, what is the causal organism for the bajra rust? What is the causal organism for the sorghum rust disease? What is the causal organism for the groundnut rust? What is the alternate host for the groundnut rust? What is the alternate host for the wheat rust diseases? I will give the one small table regarding that one. Finally, suppose if the rust disease is present, generally what type of fungicide is required for the management of the rust disease? I will give the 2 to 3 fungicide names that will be helpful to you to understand that for the management of the rust disease, we have to spray the so and so fungicide. That is the common thing. They are helpful for your examination point of view for the match the following. Definitely, they will give the time rice blast, rust disease, smut disease, and also some other fusarium wilt like that one. In opposite side, they will give the names of the fungicides. We should also match the following things. At the time, definitely you have to know about the particular disease and also what is the particular fungicide is required for the management of that particular disease. There is a chances to ask match the following. So most of the students, they lose the marks during the type of that if the questions are asked by the examination teacher. Don't worry about that one. I will also. Should we prefer other study material in addition to Angro notes for the plant pathology, sir? Okay, right. Now we have asked the, in addition to the Angro means mostly, that means Angro and also TNAO materials, more or less they are similar, but mostly they are completed with that one. That means 80% of the syllabus, it is covered in that one. For example, next class I will explain that one regarding to the one of the disease in the rice crop. Generally, in the textbooks, they will mention about the disease color. For example, if it is the reddish brown in color, But in some textbooks, instead of the reddish brown color, they will say some other colors. So if you go through all these the textbooks means definitely you will get the little bit confusion. You are asking not only I am following the Angro material. Now what I have told that one, what type of the similar symptoms are produced in the rice plants along with the rice blast means brown spot lesions. They will also give the little bit confusion. But here in Angro or TNA material, they have not covered the points. Actually, the points it was given in some other materials, but the points they are very clear. They will also give some helpful to us because of I have taken the points from the International Rice Research Institution. Okay, do you understand that one? Yes, there is a possibility to give the little bit confusion between the blast and also brown spot. Yes, that is the correct thing. Because of myself also, while I was in PhD courses, at the time also I have got the confusion exactly what is the reason for that one. In textbooks they are saying it is caused by the blast disease. It gives the it gives the spindle shaped lesion. But here there is no spindle shaped lesion. In brown spot it gives the sesame like seed like structure. But these are not the sesame seed like structures. Myself also I have got the lot of doubts whenever I went to the field. So along with the field conditions, with my awareness, with my experience, the students how would they get the doubts regarding these things? All of these things are included, and finally I will conclude all these things. If they have to eradicate these doubts means definitely we have to supply some of the material to you. I know about that one. Definitely I will give the notes regarding to these subjects. That means for the diseases and also for the fundamentals of that one. First of all, if you want to read, you should go for the H.C. Dubey book and also the textbook handbook on an introductory mycology. That will also be helpful to you for the basic knowledge. These two books are sufficient. If you read these two textbooks, they will give some information regarding to the plant pathology. I will say one thing to you. If you want to read, if you want to clear the JRF examination, you have to complete the total textbook at least 6 to 8 times. That is one of the most important things. Okay, sorry for the interruption. So first of all, you have to purchase the textbooks related to the plant sciences. And also immediately you have to start reading that particular textbooks at least 6 to 8 times before your examination. That means before the April month. Plant breeding, genetics, plant pathology, and also microbiology. How much time it is there? You have to calculate that one. So regarding the plant pathology, we should purchase the H.C. Dubey and also the textbook introductory mycology. Whether you understand or not, it is not a matter. First of all, you have to give the one read, the second read, the third read. After completion of the 3 to 4 readings, definitely you will gather some information regarding to that particular subject. Okay. Anything else? If nothing is there means I will close the. Okay, right.